Good morning to all of you. Today we will discuss something about the major points about the water-soluble vitamins. Now, the water-soluble vitamins in the body are used as coenzymes for the enzyme actions. Now, the various water-soluble vitamins include B group of vitamins like B1, B2, B3, that means thiamine, riboflavin, niacin. Then we have pyridoxine, biotin. These are the major B group of vitamins, and also we have the vitamin B12 and folic acid. And another water-soluble vitamin is ascorbic acid or the vitamin C. Now, almost all of these function as coenzymes. Now, the coenzyme form of thiamine is thiamine pyrophosphate, and it is used for the oxidative decarboxylation of pyruvate to acetyl-CoA and alpha-ketoglutarate to succinyl-CoA. And the niacin vitamin produces the coenzyme NAD, nicotinamide adenine dinucleotide, and NADP, nicotinamide adenine dinucleotide phosphate. Now, these are all used for various dehydrogenation and reductase reactions. NADP is specifically required for the HMP shunt, and from this we get NADPH, which has extensive use as an antioxidant. Now, the riboflavin is another water-soluble vitamin, and this riboflavin is used in the form of flavin adenine dinucleotide FAD or flavin mononucleotide FMN. Now, these are also required for the various dehydrogenation reactions in the body. For example, succinate dehydrogenase requires the FAD, which is converted to FADH2. Now, the vitamin C is another water-soluble vitamin which has got significance and several roles to play. One of them is in the post-translational modification and maturation of pro-collagen to the collagen molecule. It also acts as a reducing agent for many oxidant type of drugs and compounds. Now, folic acid and vitamin B12 deserve some special discussion because folic acid is mainly used in the 1-carbon metabolism. 1 carbon units are carried by folic acid and also by vitamin B12. Now, the formild folic acid, formild tetrahydrofolic acid, methinyl tetrahydrofolic acid, methylene tetrahydrofolic acid, methyl tetrahydrofolic acid, and methyl cobalamin also carry 1 carbon units. Now, these methylene, methinyl, and formild tetrahydrofolic acid can be interconverted, but once the methyl tetrahydrofolic acid is formed, then it cannot be converted to the other forms, and so what is needed is vitamin B12 or cobalamin, to which the methyl group is added from the methyl tetrahydrofolic acid, so that the methyl cobalamin is produced. Now, if vitamin B12 is absent in the body, then this transfer of methyl group from methyl tetrahydrofolic acid to B12 does not take place, and this process is known as the And in this condition, it is known as the methyl tetrahydrofolate trap. This shows that a deficiency of B12 can also cause a deficiency of folic acid. For the proper restoration of the folic acid, we need B12. So these two vitamins have to be given together. Now the folic acid deficiency can cause the macrocytic anemia, whereas the B12 deficiency causes the megaloblastic anemia. Now folic acid is required for the DNA synthesis because the purines, the C2 and C8, that means the carbon C2 and C8 of purines require the folic acid, formal tetrahydrofolic acid and methynyl tetrahydrofolic acid. So in the deficiency of folic acid, the purine synthesis will be affected, and hence the DNA synthesis also will be affected. Now the vitamin B12 requires the two protein factors for its absorption. One is known as the intrinsic factor, and also the other one is known as the cobalamin. Both these protein factors are required for the proper absorption of B12. Now in the deficiency of intrinsic factor, the pernicious anemia will be caused. Now the methylcobalamin is required for the conversion of homocysteine to methionine. Methionine is a amino acid. Actually it is an essential amino acid, but some amount of methionine can be synthesized in the body from homocysteine. Now if B12 is not available, then the homocysteine excess takes place, and homocysteine excess is a risk factor for the myocardial infarction. So B12 has to be taken by patients who had myocardial infarction to prevent further infarctions. So homocysteine is a risk factor for heart diseases and should not be allowed to increase. Now another factor as far as B12 is concerned is that B12 is not present in the vegetables. It is not present in the purely vegetarian food, unless and until we take milk and curds. Milk and curds contain enough B12, and also some amount of B12 may be synthesized by the bacterial flora of the intestine. Otherwise, vegetarians in general have a deficiency of vitamin B12. Now it is mainly present in the non-vegetarian food, and maybe injections may have to be given to those people who have decreased level of B12 in the serum, because serum B12 can be estimated more or less accurately by the modern techniques. Now, what is the difference between folate deficiency anemia and the B12 deficiency anemia? As I told you, folate deficiency causes macrocytic anemia, whereas B12 deficiency causes megaloblastic anemia. Now in this, the DNA synthesis is delayed, whereas the protein synthesis takes place, causing megaloblastic anemia. Now another factor which has to be remembered is that in B12 deficiency, in addition to this anemic state, there is an added neurological problem because one enzyme known as methylmalonyl CoA mutase converts methylmalonic acid to succinic acid or succinyl CoA. Now this enzyme is a mutase enzyme, and this requires B12 as a coenzyme. Now in the deficiency of B12, this reaction is not taking place, and so there is what is known as methyl malonic acid accumulation in the body, and then this can go to the brain and may cause the demyelination, and this will result in the various types of neurological problems. This is a very both the sensory and motor tracts are affected in this, and so it is known as severe combined degenerative state. So these are some of the important points that one has to remember when we talk about vitamin B12 and folic acid. So this is something about the water soluble vitamins, and in the next topic which is of importance is we will take up something about the protein structure. Now proteins are made up of amino acids, as we all know, and the sequence of amino acids is known as the primary structure of proteins. So the sequence of amino acids is determined by the nucleotide sequence in the gene, and it determines the sequence of amino acids, and this is known as the primary structure of protein. Now the next higher structure of protein is the secondary structure. The secondary structure may be alpha helix, beta pleated sheet, or a random coil, or the beta band. Now alpha helix is formed by hydrogen bonding between amino acids, which are 4 amino acids in distance. That means nearby amino acids form the hydrogen bonds, whereas beta pleated sheet, the hydrogen bond is between first amino acid and maybe 10th amino acid, or the second, or with the 12th. That means a little far away amino acids take part in this beta pleated sheet formation. Random coils, of course, do not have a specific pattern of this protein structure, but all these 3 structures are important for the proper function of the protein molecule. Now this is known as the secondary structure. Secondary structure further folds itself to form the tertiary structure, and most of the proteins, all the proteins rather, have the primary, secondary and tertiary structure, and some of the proteins have the higher level of structure known as the quaternary structure. Now tertiary structure means it is the interaction between different domains of the protein molecule. Now other factors other than hydrogen bond which stabilize this protein structure are the van der Waals forces, is very important, and another one is by the electrostatic bonds, that is between the extra carboxylic acid group and the extra amino group, extra carboxylic group of the acidic amino acids, and the extra amino group of the basic amino acids can get involved in electrostatic bonds, which can also stabilize the protein structure. Now, so in this manner, and there are also the hydrophobic interactions. That means those amino acids which are neutral try to come closer, because thereby avoiding the water. So the hydrophobic amino acids try to come closer, thereby further folding the protein molecule. Now the tertiary structure has got a 3-dimensional structure. The molecule takes up the shape of the ellipsoid or spherical or fiber-like or the rod-like structures. Now the quaternary structure means polypeptides having the primary, secondary and tertiary structure, different polypeptide chains, more than one polypeptide chain. They interact together to form a dimer or a trimer or a tetramer or a pentamer. So the quaternary structure will have more than one polypeptide chain. Now the examples of proteins with quaternary structure include hemoglobin. Myoglobin has got only this tertiary level of structure because it has got only one polypeptide chain, whereas hemoglobin has got 4 polypeptide chains, and so the hemoglobin is said to have the quaternary level of structure. Now, if we consider the hemoglobin structure, then there are 2 types of polypeptide chains, alpha chain and beta chain, and there are 2 alpha chains and 2 beta chains. Now, each one of them consists of about 140 amino acids, and each polypeptide chain is bound to a porphyrin molecule, and in the center of porphyrin there is a iron atom, and hence the porphyrin ion is said to be the heme. That means the heme and the globin chain form the hemoglobin. Now, the porphyrin is formed by 4 pyrrole rings which are linked by methanide bridges to make it a protoporphyrin, and this forms the one chain of hemoglobin, maybe alpha chain or the beta chain. So like that there are 4 polypeptide chains, and hence there are 4 heme, and as there are 4 heme, the iron also, the number is 4, and the iron can loosely bind with the oxygen molecule so as to form the oxyhemoglobin. When the oxygen is removed from the hemoglobin, it is known as the deoxyhemoglobin. Now, there are 2 histidines molecules. One is proximal histidine and another one is distal histidine. Both these are involved in the oxygen binding. Now, the ion at the center of the heme has got 6 valences. 4 of the valences are satisfied by the nitrogen of the 4 pyrrole rings. The 5th valence is actually bound to the distal histidine, nitrogen of the distal histidine, and the 6th valence is free in the deoxygenated hemoglobin, but will be occupied by the oxygen when it is oxygenated. Now, the oxygen attachment to the ion is very loose, and it is not perpendicular. It is at an angle, so that the oxygen binding to the hemoglobin, the iron part, is a loose one, and if when required, that is in the tissues, this oxygen can be delivered to the tissues. Now, in the lungs, there is the partial pressure of oxygen is 100%, and hence the hemoglobin can very easily bind with the oxygen. 4 molecules of oxygen bound to 1 hemoglobin molecule, and then it can be taken to various parts of the body, and this the PO2, that is the partial pressure of oxygen in the tissues, is about 30 to 40 mm of Hg, and hence the oxygen can be released into the tissues. So this hemoglobin is an ideal oxygen transporter. No other molecule in the body can do this versatile function, not even myoglobin. Myoglobin cannot do this function because myoglobin has got only 1 polypeptide chain, and so it can bind only 1 molecule of oxygen, but the binding is very of great affinity, and so the myoglobin cannot release the oxygen at a partial pressure of about 30 to 40 millimeters of Hg, which is obtained in the in the tissues. But then what is the use of myoglobin? Myoglobin is present in the muscles, and hence it can store oxygen, and when we go to the high altitude or when we are doing heavy physical exercise, then the partial pressure of oxygen will be down to 10 or below 10, and at this low partial pressure the myoglobin can release the oxygen. So it has a function. At a very low oxygen tension, the myoglobin is able to release the oxygen for various biochemical phenomena. Now, as there are 4 polypeptide chains, there is cooperative binding between these polypeptide chains of hemoglobin. meaning that if we plot a graph of oxygen, rate of oxygen dissociation or a rate of oxygen association, and the partial pressure of oxygen plotted on the x-axis and the association or dissociation of oxygen plotted on the y-axis, then we can see that as far as the hemoglobin is concerned, it gives a sigmoid or the S-shaped of graph, whereas in the case of myoglobin it is a hyperbolic graph obtained. The reason for this difference in the binding of oxygen is that the binding, as far as hemoglobin is concerned, the binding of one molecule of oxygen to one heme of one polypeptide chain increases the affinity with which the second molecule of oxygen can bind with the second molecule of heme and the polypeptide chain. Now the second molecular oxygen binding to the hemoglobin further increases the binding of the third, and it still further increases the binding of the fourth molecule. So initially the oxygen binding is slow, but then it picks up fast because the affinity is increased. This is what is happening in the lungs. But in the tissues, the reverse is happening, where the oxygen, partial pressure of oxygen is quite low in the metabolically active tissues. It may be around 30 to 40 millimeters of Hg, and the release of one molecule of oxygen from hemoglobin decreases the affinity of oxygen binding to the second, third and fourth heme molecule, and hence the second, third and fourth molecule of oxygen are released rapidly, and so the release of first oxygen molecule from hemoglobin helps in the release of the second, third and fourth molecule of oxygen in the tissues. Now, so this is a advantage with the hemoglobin, where both the binding of oxygen and the release of oxygen or the unloading of oxygen is very effective. Such a function cannot be attributed to the myoglobin, which has got only one polypeptide chain. Now the presence of 2,3-bisphosphoglycerate also helps in the unloading of oxygen. 2,3-bisphosphoglycerate is a byproduct of the glycolysis in the RBC, where the 1,3-bisphosphoglycerate is converted to 2,3-bisphosphoglycerate. In this process, one ATP is also lost. In the normal cells, 1,3-bisphosphoglycerate becomes 3-phosphoglycerate, where there is a substrate level phosphorylation taking place and ADP is converted to ATP. But now, as 1,3-bisphosphoglycerate is converted to 2,3-bisphosphoglycerate in the RBC, especially in the RBC, there is no ATP production when the 2,3-bisphosphoglycerate becomes the 3-phosphoglycerate. So there is a wasting of energy taking place in the RBC, which is actually required, because the RBC does not require much energy, because there is no nucleus, there is no DNA synthesis, there is no protein synthesis, or no major functions are going on in the RBC. Only it happens to be containing hemoglobin, which is a transporter of oxygen molecule. So high energy may be rather deleterious to the RBC. So it is taken care of by the formation of 2,3-BPG, and thereby wasting the energy. But 2,3-BPG has got a very great importance in the unloading of oxygen, especially when we go to high altitudes, when there is a hypoxia, tendency for hypoxia present, then the 2,3-BPG concentration increases, and also the pH pH is decreased, and hence more oxygen can be made available. Now, as and when the oxygen is released from the hemoglobin in the tissues, the hemoglobin can bind with the carbon dioxide to form carbaminohemoglobin, and this carbon dioxide can be taken back to the lungs for expiration. So hence, in the tissues, the increase in the concentration of carbon dioxide and decrease in pH will help in the release of oxygen and binding of the carbon dioxide. This is known as Bohr's effect. The reverse happens in the lungs, where the oxygen concentration is very high, so the binding of one molecule of oxygen to the hemoglobin will release the carbon dioxide molecule and helps in its exit from the lungs. So this reversible phenomena is known as the Bohr's effect. So in this manner, hemoglobin is one of the very important proteins. Very salient features are present in it, and very dynamic molecule.